let me start first, and then I will hand over the later part to Mr. Telmo and Caroline. Today we are going to cover two parts. First part is about the product context about our MyLab BCM. So through this session, we are going to go through why CBC flagging and morphology need a single governed workflow. And second part is about the Joel Briggs case study. We welcome Mr. Joel Briggs for today's guest speaker. He's the NHS blood science lead in Western RS field, and he will share his experience through operating context, service challenge, remote morphology review, governance, and real-world value in the Western RS. So firstly, we are going to go through this product context first. So we are going to introduce our product first and then move to Joel's sharing of his real-world experience. So before we move on to MyLab BCM introduction, I just want to touch about the NOUL. We established our product, our company in 10 years ago, December 2015. Our name is NOUL. So it was founded by our CEO, Chan-Yang Lim. We have been invested about $85 million up to now. And today we are having 110 persons of employees. We are located in Republic of Korea, and our key products are blood analysis, like CBC and morphology for this BCM, and malaria diagnosis and cervical cancer screening tool. So these are our photos of our headquarters research lab and plant in Republic of Korea. Our mission is to explore global challenges that threaten human health and life, discover novel potential solutions, and realize those possibilities. As I mentioned earlier, we have three products as known as MyLab. First thing is malaria diagnostic solution called as MyLab MAL. It is about the digital microscopy in line with the WHO gold standard, gemcitabine. And also we have through this MyLab MAL, we can suggest species, positive, negative, and falciparum and vivax and others. And also we can analyze life cycle stage basis for blood stage malaria parasites. And finally, we can share remote readout using high definition digital images. For today's blood analysis solution BCM, it is about single run CBC and blood cell morphology from just 5 microliter of whole blood, and integrate WBC double differential and digital smear interpretation. And finally, we can also share the remote readouts using high definition digital images of cells. And finally, we have cervical cancer solution, as known as MyLab Cervical. It is about the automated pap staining, digital imaging, and AI analysis solution. And also we can processing sample support from the FBC slide and minimize screening times for users. And finally, it also can share remote readout using high definition digital images. Today, we are going to focus on the MyLab BCM from now on.
Sophia, sorry,
yeah,
we are back.
We have Tamo again, so please share this part.
Yes, so like Sophia was already showing, and has all the ones that are online, the blood test workflow is following what is described in this slide. So the hospital, the physician will order the testing, then the laboratory will perform the test, will do the first screening with automatic analyzer that is installed in your laboratory. So the flags, it will show the need of review. So then a smear is performed, the staining is performed, and then comes the morphologist personnel to review under the microscope. And then finally, we will have the final report and the results validated to send to the patient. Next slide, please. So what we see in this constellation and all this workflow common everywhere across the globe is there several gaps that can be closed using our device and our technology. So we are talking about the smearing process that in much of the cases are done manually. We are talking about the staining process that generate lots of waste, is a time consuming, and it's pretty much experienced person dependent because the variability of the staining is huge between the lab technicians. And also we have the first analysis that also requires an expert to check and to classify the cells. So this is where the MyLab can be placed and bring add value, improving your workflow efficiency. Next slide, Sophia, please. So in these scenarios, and just give an overview about these limitations in the current workflow. So every countries went the process of centralizing in mega laboratories the samples. This creates problems about increased cost of transport, about degradation of the samples, representing delays. Also, extending the turnaround time for results. And of course, that is a global issue to have hematology experts available to go microscope and to classify the cells and make the final validation. Next. So, next slide, please. Yes, so MyLab, a digital hematology platform that can enhance the blood testing and innovative technology can be used to, next slide, to standardize, to bring to life everything that is connected with manual workflow that we described before. Please, next. Oh, so all the manual smearing, so that from the sample loading, that is completely simple. With 5 microliters of blood, you will place the sample in our cartridge. The cartridge will be introduced in the device. The device will perform the smear, the fixation, and the staining, a full automatic process. Then optical imaging is starting to collect all the images, and afterwards on-device AI analysis will deliver the results on screen. So this is the intelligent automated workflow without any manual work, guarantees standardization and traceability for all the process. Next slide, please. So what I want to showcase here with this slide is that our cartridge is a innovative patent medical device where the stainings are in a solid stain. So it's the next generation staining and immunostaining where the new methylene blue, where the eosine and the buffer are in solid stain. So everything that is from the reagent preparation, methanol fixation, drying, staining, washing, drying, the manual process is completely void. So we don't need any type of these steps manually to be in place. And also, it's a methanol-free solution based the fixation on ethanol. So the cartridge will perform the slide, will stain, and also deliver the analysis afterward. Please, next slide. So all this manual works that was done and required with the flag generation from the hematology analyzer will be avoided. So this is a unique solution to small, medium-sized hospitals where the CBC and morphology analysis on site, it's a need, where a point of care testing to decentralized measurement is also a need to bring the diagnostic closer to the patient and minimize the volume of blood that is necessary to have a full report. It's 5 microliters of sample comparing with other products range that needs 15 to 1 milliliter of sample to aspirate to deliver the full results. Next slide, please. So, and you can ask, okay, how the device correlate and is in agreement with the reference devices from a laboratory nowadays. So here I can give you some numbers about correlation regarding the CBC parameters compared with the reference analyzer installed in the laboratory. A NOUL team always use gold standard devices to make validation and correlation about our parameters. This device is different from the one that Joel used in his laboratory that we will present later on during our webinar. But here you see a correlation bigger than 0.95 for all the basic parameters from the CBC. And in the next slide, you see the full agreement about the differential and also about the flagging. So you see a differential with a very good correlation. You, of course, basophiles, eosinophiles decrease the correlation because the number of cells is very low. But the most important thing is the agreement between the flags regarding blasts, abnormal lymphocytes, and atypical lymphocytes. So I can tell you for the experience that we have now that our device is pretty much accurate with high sensibility and high specificity to distinguish a reactive situation from a non-reactive situation that in a decentralized scenario is pretty much important when and where to refer the patient in these two types of health disease and situation. Please, next slide. So this image that you see here, they are not treated with any kind of software to manipulate the quality of the image. These are real images that you will find in our device that will be available also in the viewer, where you can see the quality of the staining, where you can distinguish the different type of nucleus, where you can evaluate the granulose, and you can see also the chromatin. So these are the images that our AI also use to distinguish a blast from a typical lymphocyte, guarantee that the non-reactive and reactive situations are completely well classified. Next slide, please. So connected with our device, as I said before, we will work with a software viewer. So this viewer is software that can be installed in all the workstations in your laboratory that will receive the results from our MyLab PCM device. And in this viewer, the user will be able to reclassify, drag and drop the images in the different subcategories, can also be able to personalize reports with the name of institution or hospital, with the name of the lab director, writing comments, and of course, generate the PDF to send it across the departments. This viewer can be installed in any workstation without cost of the license. So we don't charge any cost for any license of the software. Next slide, please. Regarding validation verification, so we are CE marked, we are IVDD, we are in the process to receive IVDR, but nevertheless, our potential users want to have their own evidence about the quality of results. And in several places, from Netherlands to Germany, from Korea to Indonesia, US and Kuwait, who have validation process in place, with different analyzers and always with the gold standard and reference method in place that will deliver the correlation and also the agreement of the flagging. As soon as these studies are ready, we will share publicly all these results and also the publication that comes from here. Next slide, please. Next, yeah. So when you think about different scenarios where the MyLab can bring add value, increasing efficiency in the workflow, decreasing turnaround time, time for analysis, decreasing costs, we can think about a satellite laboratory where it's already in place with the normal hematology analyzer, but doesn't have any morphology available, where the experts are not available. This is one of the scenarios that the MyLab can add value. Also, GP clinics, emergency rooms, intensive cares, where the results are needed in loco, so the numbers plus the analysis of morphology, and also in pediatric settings. So the pediatric settings are important for children to receive immediately treatment. We are proceeding a validation of our device for pediatric samples below 3 years old that will be ready before summer this year, and connected with this, also the capillary blood measurement, it will be available at the same time. So we know for children in point of care settings, the capillary fingerprint availability application is one must to have, and we will deliver that in a couple of months. Next slide, please. Sorry. So after all this product presentation, and I'm really apologize the technical initial problems with the internet connection, but my backup from the HQ, Sophia, handled that perfectly, I'm sure. We will go now through a second part of our webinar. So in this second part, is next slide. We will cover the following. So we will introduce our the Joel Briggs from Western Islands in Scotland that perform evaluation. So we will share the full context about this. What was the challenges? What was the findings comparing our device with the reference device from the laboratory? And also, what were the benefits? I hand over now to my colleague, Caroline, that will give a short introduction, and we take it from here.
Thank you, Telmo. Thank you. Hi, everybody. Nice to meet you. So, Joel, I'd like to say thank you for joining us. We had the pleasure of meeting Joel at IBMS last year in September. So thank you for joining us. Before we do dive into the technology, can you briefly describe your role to us and to the audience out there, and the setting that you work in, especially for those that may not be familiar with the Western Isles and the challenges it may offer?
Yeah, thank you very much, Caroline. So I work as the laboratory services manager for the supporting laboratory services over the entirety of the Western Isles. Geographically, it's a very unique environment. We have multiple sites across multiple islands. There's long distances between us, very limited transport options, which are weather dependent. Some of the sites can be separated by hours or potentially days if the ferries and flights are disrupted.
Yeah, so next slide, please, Fia. How does geographically this affect the laboratory service in practice? So in real world, what you are facing concretely in this type of geographic setting?
So our geography has a huge impact on our service. We only have trained morphologists on one of our sites because the other two sites are very small. And moving samples or staff between these islands is very slow or expensive, and sometimes impossible to do at short notice. However, the clinicians and patients still need a timely and reliable service, and especially for urgent cases.
Okay, so I guess this is where digital morphology becomes really relevant within our offering. Can you explain why remote access is so important within your setting?
So remote review is really absolutely critical for us. With digital morphology, you can prepare a smear on one island and then be reviewed by an expert on another island or even potentially the mainland if we need to refer a case on to a consultant for review. This means the clinical decisions aren't being delayed by our geography, and the staff on smaller sites don't feel like they're working in isolation.
Yeah, so I think this is really quite evident. So talking about analytical confidence, you compared the MALA-PCM results with your routine analyzer, a Beckman Coulter DXH 800. How was your finding? So how important this was for you and for your team?
So getting a good comparison was absolutely essential for us. The DXH 800, as you say, is our established reference method for CBCs and flagging. So anything that we bring in alongside this has to align well with it. What we observed was a really strong correlation between the DXH 800 flags and differentials and what the BCM was showing us at the morphology level.
Okay, so go a bit more diving through the numbers and what we saw regarding this evaluation and what you shared with us. So we see a good agreement with all the clinical parameters regarding correlation parameter and also the flag agreement. I also emphasize that I saw that the Beckman Coulter was a bit in a safe way, generating a bit more false positives that the NERBC were not present when we saw in the microscope. So in practical terms, what strong concordance mean for your team? So what this brings really for your team?
So really, it meant that when we were getting a result from the DX800 that would raise a flag or a concern, the BCM was consistently supporting that with the visual and morphology examination. Equally important, we didn't get anything coming up on the BCM that was unexpected and discrepant from what we were getting from the DX800. The system behaved in a predictable and clinically coherent way, which is exactly what we wanted to integrate a new piece of technology.
So did that help with the clinical acceptance, of course?
Yeah, absolutely. So once our staff witnessed that the BCM was giving the same results that the DX800 was giving, the confidence in it really grew quite quickly. And I think being able to see the same cells on the screen as you were seeing down the microscope really increased that confidence. The BCM really felt like it was an extension of our existing workflow rather than sort of competing or giving us contradictory information.
Wow, that's brilliant. And how important was that image quality in establishing that trust, seeing as we are newbies to the market?
So image quality is absolutely fundamental. So for remote review, you need to be confident that what you're seeing on the screen is basically what you'd see if you had the microscope in front of you. And with the BCM, the resolution and consistency allowed us to basically make the same decision we would do with just a microscope.
Amazing. And then some people, I guess, would worry that remote review could reduce safety. What's your experience within this matter?
In our case, it's actually improved safety. So with the traditional method, a single person could be making a decision in isolation. With the BCM, we have the ability to share that review. We have full traceability and full auditability. The AI that's in the system helped to triage and speed up the process a bit, but it's still very important to us. There's expert overview there. It just means that some of our physical location difficulties are removed.
Thank you.
So how this model can be used and it will be used to affect the workflow force remain in the remote region. So how the MyLab BCM it will support this lack of staffing.
Right, so I think for us it could be extremely transformative. Recruiting, retaining experienced morphologists in a remote area such as we're in can be extremely challenging. The digital morphology allows us to share some expertise across sites, also supports training and reduces staff burnout. I think it will make our service far more resilient.
And do you see this supporting decentralized or near patient testing?
Yeah, I think it will, provided that the governance for this stays within the pathology lab. Decentralization without the digital oversight is quite risky, but with the digital morphology and remote review, I think it's safer than our current traditional method.
So if I challenge you now to give three words that can describe our system, what could be these three words that easily describe the MyLab BCM?
So I'd say consistency, confidence, and equity. So consistency, knowing that we're getting the same results as we would with our DX800s, confidence in the decisions we're making, and equity because we're able to provide equitable care for everyone across our islands, no matter where they're living.
Amazing. So, Joel, I would just like to say thank you. It's been a really good kind of first powerful example of how digital morphology supports both analytical rigor and real world service delivery in challenging geographies like yourself. So, thank you very much for sharing this with us.
Yeah, thank you, Joel. Thank you, Caroline. So I'm challenging now all the participants to address all the questions, all the queries, or clarifications that I need in our chat. My colleague Dave, my colleague Caroline will support in all the queries. If we don't have time now in the next 15 minutes to answer all the queries, our team will collect all of them, prepare answer, and will share with you by email. Nevertheless, after this webinar, we will also send to all the participants the record version of the webinar, also the presentation that can anybody that was not able to attend can attend. And as well, I will really invite you to answer our survey at the end of this webinar, because your feedback is important for us to improve and to change, if you see it, the future webinars that NOUL will deliver in a short term. So first question, if we have any questions in our channel or in our chat. So far, I don't see any questions, but I can tell you that we received pre-questions before. For example, the ones that were related with new testing and availability of new testing and parameters that are clinical relevant for the MyLab. And I can disclose, so at the moment, we are pretty much focused to deliver the good quality screening for reactive, non-reactive situations using the clinical parameters available for 5-part differential CBC countings, but reticulocytes count and other type of tests, morphology, red blood cell morphology, based in inclusions, based in shape. It's in our roadmap for short-term development that we expect to be available. So other question that we receive, it's about if the device can process how many images per sample we can process. So we after we find and stain our smear, the device will collect more than 400 images that will be used by the AI to classify as much accurate as possible the cells that are present in these samples. Nevertheless, this is a non-ending process. So we are continuously receiving data based from our evaluation sites across the globe to continue training and improve the sensibility and specificity about our morphology classification. Yes.
Shall I ask another question? Yes. So there's some questions that come through. One was, what is the scope and nature of the hematological cell data used to train the MyLab BCM AI model? So could you go through that, please?
Yes. So the scope that is based it's based in real cases. So we are collecting real cases, clinical cases from our scientific partners across the globe, hospitals, private clinics, private lab chains, that are sending us the cases that we send to our R&D team in HQ in South Korea to train the AI. And also we use the clinical standards images available that are used in general to train AI devices for diagnostic purpose. So that's how we do it.
Brilliant. So I've got one more question here that's just popped up. How does the MyLab BCM manage or compensate for variations in staining quality to maintain consistent and accurate results?
So the device is use properly algorithms and score levels that are connected with characteristics of the cells to compensate the changes and minimize them. So we don't see absolutely variability between the staining process in different cartridges, it's different samples. So we guarantee a standardized process of staining and a standardized process of analysis. This is pretty much what Joel described about the consistent results that are required in these decentralized settings.
One more has just popped up, and this aligns well with our visits to the large hospitals we've visited recently. Beyond FBC and morphology, does the MyLab platform offer analysis for other parameters, such as platelet clumps?
Yes, yeah. So what I can disclose to you, and we have some attendants that are already, we talk and discuss, we saw that our MyLab device, in the presence of PLT clumps, deliver accurate PLT counting. Why? Because as we go to the microscope to estimate the PLT count, also the MyLab count the platelets present in the clumps. So we deliver the flag, but we also deliver accurate result. So in hospitals like Eurofins in the Netherlands, that is a private laboratory, but is placed in a big hospital in Apeldoorn in the Netherlands, like BCRT in Berlin, like Oxford, like St. Thomas in the UK, they see a really clinical value to use the MyLab BCM as an alternative method when the normal conventional hematology analyzers deliver a pseudothrombocytopenia with a clump flag. Then you don't need to estimate, or you can deliver accurate PLT counting using the MyLab, not stopping surgical procedures when you don't deliver the PLT counting.
Okay, we have some questions. We have some live questions that are appearing. Let me just see if I can see them. Okay.
So
this is one. Sorry.
Yeah.
Go ahead. This is one for Joel. Joel, if you can hear me. In which dear patient cases do you see the big benefit of using the MyLab BCM, and which diagnostic issue?
So I think for us, one of the big benefits is, again, comes back down to our geography, really. That we really struggle with getting samples from our furthest down island, which is Isle of Barrow. That can sometimes take over 24 hours to get a sample up to us, which obviously, when you're looking at blood films, the cells have really started to degrade. So having the ability to get a smear made where the patient is, rather than moving the sample up to us, will be hugely beneficial.
Okay. Which diagnostic issue? That makes complete sense. So did you say we have 2 or 3 more questions? Can you see them on your chat? No. Here we are. Let me just open them. Here we go.
Yes.
Okay, another one. How does MyLab compare to other point-of-care devices at point-of-care settings? Good question.
No, this is a very wonderful question, because we don't compare, because the real point of care device regarding the MyLab doesn't exist. So we are unique in our approach, because we are the only one that deliver the full count, blood cell count, 5-part differential in morphology analysis. If we compare numbers with numbers, we are completely aligned with other solution that is in the market that only deliver numbers. If we compare with manual microscopy, we are 100% comparable with manual microscopy. But in the market, combining the 2 solutions, numbers, flagging, and also microscopy analysis, we are completely aligned with the reference method that is described in the CLSI guidelines.
Perfect. And this is a wonderful question because exactly where we're targeting. Can it be used for oncology patients?
Yes. So this can be used for oncology patients, special that are the ones under chemotherapy. So in this situation, before addressing the therapy, a neutrophil count is always needed. And this is where we see also head value and the benefit having the MyLab to measure immediately close to the patient the neutrophil count to decide to address the chemotherapy or not.
Let's put down so more down here, move that little thing.
Mhm.
How can we share BCM results at the moment?
So the results can be shared at the moment using the intranet from the laboratory. So it means that the workstations, they need to be connected in the same IP as the viewer and the MyLab. However, by the end of this year and during this time, we are developing the cloud-based connectivity that will eliminate this situation where any workstation across any network can connect remotely to the cloud server to receive the results.
Thank you. And how can the AI-assisted morphology flag abnormal cells like blasts?
So the AI assists identifying them and counting them. So this is what Joel referred about the agreement with the flag from the reference analyzer. So the difference is regarding the flags, you in a normal conventional analyzer, you receive a flag and you have a scattergram, and you need to imagine a blast cell positioning in that scattergram. In our device, you have a blast flag. You click in a blast flag and you receive the image that generates the flag, and you immediately can see and reclassify if it's necessary.
Thank you. We have a question from Tajish. Will you rise or develop NRBC consistently with staining or software updates? Normal blasts have bad correlation and crucial to determine them in remote locations when a patient is dying. Need urgent response.
Absolutely. So we have a very good correlation with NERBC counting already, and as well blast detection. So it will be available always remotely. So our staining, right, GIMSA staining, is the one that is used to classify and to count NERBCs and blasts.
Perfect. And have we compared the performance of MyLab among patients with blood stage pathogens? Hannah has asked.
So you were talking about pathogens like infections, I believe. So what MyLab do, and Joel, I think you are in agreement with this, we cannot say if it's a bacterial infection, we can say it, or viral, but we cannot say what bacterial is inside or what virus. So we can distinguish between reactive, non-reactive. Reactive, we can say it, okay, could be the high probability to be a bacterial infection because of all the neutrophil related parameters are high, or could be a more viral infection. But we don't have the level of discrimination to identify the pathogen that is present.
Thank you. And what about QC? What do we have?
So QC at the moment, we will have the 3 levels QC available that can be handled as a normal patient sample where you will receive your assay, your target value, and also available the Levy-Gini graphs to evaluate the trends and see the performance of the QC. Yeah.
Okay, thank you. We can send the results in PDF. Yes, we can. I can answer that one. Thank
Yes, please, you can.
And has the system been compared to doctors' manual interpretations, or is it sensitive and specific, and is it very reliable?
Yeah, so I think Joel can give a testimonial about this question. So, Caroline, can you repeat just to be more precise?
Yes. So has this system been compared to doctors' manual interpretation? So I guess it means by looking down, has it been compared to manual microscopy? And is it as sensitive and specific as looking down the microscope yourself?
You are with you, Joel, yes.
Sorry, I just take myself off mute. So It's compared to our on-site microscopy. So and yeah, when we've done things like manual differentials and compared them with the MyLab results, we've got very similar results.
So the way that you see the microscopy morphology is also pretty much aligned how the AI at the moment is trained to identify these abnormalities as well, correct?
Yeah, yeah.
Okay.
I think that's it. That's all our questions
Yes,
for now.
yeah, we have all the questions, but again, this is not over here. Please address all your questions in the channel. And I really ask you a huge effort. After you disconnect, you receive a pop-up a window where you deliver your feedback and where you will address your suggestions for approval for the next webinars. Joel, I really want to give you a big thank you to be available with your busy time and lack of resources that you need to cover the majority of the time. It was tremendous effort from our side and from your side and your team. Hopefully, the MyLab will showcase in different areas and other colleagues will come to our webinar sessions to testimonial the benefits and add value of our solution. Caroline, thank you for being with me.
Thank you, everyone. Thank you, Telmo. And we'll say goodbye.